This was the last question that we had done. Let's move on. It's the same question that's continuing. So they're now saying that phenyl, I mean, whatever this molecule is, okay, this molecule over here, it's complete the table about the reaction of with the three reagents, sodium and the structure of the products. So I'm going to draw and sketch the products on this thing. So remember, with sodium, what's going to happen is that phenols are acidic. Okay, remember that. Let's open the board as well simultaneously. And we did the comparison of acids. So I mean, this was electrophilic substitution comparison, and then somewhere we did, this is hydrolysis, this one. We did the comparison of acids, and we said that phenols over here, OH bonded to benzene, that is acidic, and that is acidic because it reacts with sodium and sodium hydroxide, but it doesn't react with sodium carbonates, which are weak bases. So it is acidic, but less acidic compared to carboxylic acids. And we did the reactions as well. We talked about phenols being acidic, and why were they acidic? Because the lone pairs on oxygen over here, it would eventually get delocalized with the benzene pi electron cloud, which is why the negative charge density would be lesser and it wouldn't attract the H plus one ions. So the H plus one, there would be more chances of the H plus one breaking off. So with sodium, the reaction would simply be that the H over here in this molecule, uh, it would become O minus one, and Na plus one would would join with it to form a salt. This is what the product is going to be, and it's going to be a redox reaction. The type of reaction, it's going to be a redox reaction. Remember that metal plus acid reactions, they produce salt plus H2. They're always redox reactions. Why are they redox reactions? Like if you take any acid, Na plus HCl, it's going to form NaCl plus H2. So what happens to Na in this case is that Na was neutral initially, और बाद में जाके it ended up turning into Na plus 1, which means it's getting oxidized. And H was actually plus 1 initially. It was an acid, HCl. And at the end, it's changing into H2 gas, which is neutral. So metal plus acid reactions are always redox reactions. Okay, ye yaad rakhna. The next, they're talking about the reaction of the same thing with with excess bromine aqueous. So पिछला rub off कर देते हैं. Now with phenols, what's the reaction of phenols with bromine? Uh, the reaction of phenols with bromine is this one. So the reaction of phenols with bromine. Let's talk about the electrophilic substitution. Where's the, just wait 1 second. This one. So electrophilic substitution. Remember under 246 directing groups, this is hydrolysis. So 246 directing groups, we studied that that you had phenols and you had phenylamines, and the benzene electron cloud was highly activated, which meant that what that meant was that the lone pair would be overlapping with the pi electron cloud, and the electron density would be very high at the 246 positions. So bromination would happen at a very fast pace. There would be no catalyst needed, and at all three positions, bromines are going to get added. And the same was true for nitration as well, that if a phenol was to undergo bromination or nitration, NO2 would get added to all three positions. So इसमें क्या होगा कि there would be uh, I mean, this is OH, this is position number 1. So bromine will get added over here at position number 2. It would get added to position number. It would also get added to position number. I mean, this is position number 2. This is position number 3. Then this is 4. No bromine would be added at position number 6 because there's already a bond over there. So nothing much would happen at that particular position. So that's That's uh, that's the bromination of phenols that is happening over here. Ali, is this clear? Sarah, is this clear?
Yes, sir.
Yes, sir.
I said the next part is uh, excess hot NaOH. What does that do? Now, excess hot NaOH, what that would do is, and remember, it's the two things that would happen. One is that phenols are acidic. So NaOH would definitely go and react with the, I mean, they're going to go and react with the phenol part to form a salt. So hot NaOH is being used. But the other thing is, you should also notice that there is an ester link, an ester group. So I'm not going to draw the whole molecule. Think of the benzene as R groups. There's an ester link in the middle. So the other part that's going to happen is that esters or amides or any of the condensation polymers, they hydrolyze when you heat them with an alkali or with an acid. The ester link would break, and this side would become a carboxylic acid, and this other side would become an alcohol. And since the hydrolysis is alkaline, the acid would not be formed. The acid would react with the alkali to form a salt. So instead, you're going to get a salt and an alcohol. So think, I mean, what you're going to do is you're going to split the molecule in the middle. Okay, there would be two products that would be formed. The molecule would split in the middle. This part over here would be gone. This would be the carboxylic acid, except that under alkaline conditions, it would actually form a salt. And this would become your alcohol part. So it would be, so the middle part is gone. The two parts would have broken apart. So if I were to draw the two molecules, well, I've drawn the molecules over there. You can have a look over there. Ariba, is this clear?
Yes, sir.
ठीक है तो इसको जो नाम दोगे ना this would be called I mean the type of reaction would be hydrolysis. ठीक है hydrolysis is happening. So remember esters and amides, they all break down when this happens. Next one is, what is the Bronsted-Lowry definition of an acid? It's a proton acceptor, that's it. Or anything that accepts H plus one ions. Now, about amino acids, draw the structure of the Zwitter ionic form of glycine. Now, we did not do amino acids, so I'm just going to quickly cover a bit of amino acids. I think we did polymerization, and we talked about polyamides, and we talked about polyesters in the last class. So I'm just going to cover amino acids now. And we did talk about the properties of polymers, and we talked about side chains and cross linkings, and the presence of different groups and the intermolecular forces between polymers, and what effect does that have on the properties of the polymers. Now, just very quickly, we'll talk about amino acids. Uh, Amino acids are a type of polyamide. They're called polypeptides. Which are basically the same as polyamides. They have amide linkages. So that's the name. What's an amino acid? Amino acid is the basic building block of your of any living organisms basically on planet Earth. Everything is pretty much made out of proteins. Uh, so amino acid, the basic structure of an amino acid is that it has a carbon atom in the middle, and there's a carbon atom that's connected on two sides with two groups. So the two groups attached to it. There's a carboxylic acid group, which is known as the C terminal, and there is an NH2 group. So there's an acid group because it's carboxylic acid, and acids can lose their H plus 1 ions. So this H plus 1 over here can be lost by this amino acid. And on the other side, you have a basic group. The N has lone pairs. Remember, anything with N that's basic, except for amides. So this side is basic, and then there would be two other things attached to that carbon atom in the middle. Uh, there's going to be an H, and there would be some R group that would be attached to this amino acid. That R group, all amino acids have a different R group, and all those R groups are given in the data booklet. Most of them. I mean, there are around 19 amino acids or 21 amino acids that make up the human body. Uh, so if you open the data booklet, you'll you'll be able to figure out the names of many amino acids. The only difference in each amino acid would be this R group. That is going to be different. So just a second. Acha, where's they note this? This over here. So each amino acid, I mean, these are the R groups that are given. You could have a CH3, you could have a CH2COH, which is aspartic acid. All amino acids are known by a single letter. There's a one letter symbol and there's a three letter abbreviation for that as well. Alanine would be, the R group would be CH3. Okay, most of you have done bio, they pretty much know all of this. So in alanine, this would be CH3. So you'll call this alanine. And the R group would be different for other molecules. For example, glutamic acid would be CH2COOH. So the R group would keep changing depending on the amino acid. And most amino acids, they are going to be chiral. They usually chiral except for one. I mean, this carbon is chiral. Chirality is when the carbon would exist as mirror images. There would be two non-superimposable mirrored versions of each other. So there are going to be two versions of this. Each amino acid is going to have two versions, except for one where the R group is H. For chirality to happen, you need the carbon atom that has four different groups attached. So there's only one amino acid that is actually not chiral, and that's glycine. Glycine over here has H as the R group. So that is the only one that's not chiral. अच्छा और क्या है कि about amino acids and polypeptides. The other thing about them is, yeah, the clear. Is this clear, Sarah? Aisha, is this clear? Abu Rera, clear?
Yes.
Yeah, sir.
अच्छा अब उससे आगे क्या है? There's amino acids. All amino acids, they have this thing that's different. Amino acids have three types of structures. The first one is a primary structure. ठीक है? All this information is stored in the DNA. Every structure in the human body, like if it's a cell membrane or I mean anything, everything is made out of amino acids forming a polymer. So there's a primary structure of a protein, and this one tells you the sequence of amino acids, which amino acids would be connected if the human body is making a hair or it's making skin or it's making a red blood cell. The sequence of amino acids and the way they're going to be connected, that's going to be different. So the primary structure tells you the sequence of amino acids, which one comes first, which one comes later. And there are 20, 21 different amino acids. Uh, every time the sequence is going to be different, and all that information is stored in the DNA, which is copied onto the mRNA, which then synthesizes a protein molecule. But that detail has now been excluded. Okay? So you're going to see past papers where mRNA and DNA and synthesis of protein is mentioned, especially before 2016. So ignore that. That is no longer in your course. You just have to know what the structure of a protein looks like. So it's usually from N terminal to C terminal. For example, they might give you a molecule like this. Like there's ALA, then there's GLY, then there is GLU, then there is ASP, and then there is, let's say, there's a small polypeptide, uh, and there's VAL. And finally, you have your C terminal. Now, what's the N terminal and what's the C terminal? Now, the N and C terminals are simply the way you're going to put the amino acid. Like an amino acid has a basic group on one side and an acetic group on the other side. It has a carboxylic acid group and an amine group on the two sides. The molecule could be put the other way around as well. Like the carboxylic acid might be facing the left side and the amine group would be facing on the right side. That would create confusion. So just to not have confusion, the NH2 part is always kept on the left side. That's the N terminal side. So you're going from the N terminal to the C terminal. And so I'm going to draw the alanine first. And you'll be asked to draw these polypeptide chains. The first one is, now all amino acids would look exactly the same. There's going to be a carboxylic acid, and there's going to be NH2. The only difference is what's attached on the fourth side. So I'm going to draw uh, the other one is GLY, so I'm going to draw that.
So इनके R groups तो दिए होंगे ना क्वेश्चन में.
नहीं, R groups नहीं लिखे होंगे क्वेश्चन में. डेटा बुकलेट खोल के देखने पड़ेंगे, ठीक है? तो भी देख लेते हैं. तो I'm just going to draw one of them, ठीक है? इसको कितने ड्रॉ करने? 5. There are 5 amino acids. So NH2 और 4 हो गए. कितने? एलेनिन, ग्लाइसिन, ग्लूटामिक एसिड, एस्पार्टिक, वालिन, ठीक है? So there's one more. ठीक है? आप लोगों को जो क्वेश्चन आता है उसमें यूजुअली सिर्फ 3 होते हैं, ठीक है? अच्छा, now the R groups. I'm just going to copy the R groups from the data booklet, ठीक है? So first one, alanine. What's the R group? R group. Alanine is CH3, so that is CH3. So I'm going to put a CH3 over here. What's the other one? The other one is glycine. Glycine, I think, was H. ठीक है? Just H. Then you have glutamic acid. Glutamic acid was what was glutamic acid? CH2, CH2, COOH. So that was CH2. Then another CH2, and then finally you had COOH. That was glutamic acid. Then you have ASP, which is aspartic acid, which I think is just CH2, followed by COOH. And the last one, aspartic acid, we देख लेते हैं, check कर लेते हैं. Valine, aspartic is this one, yes. Valine is at the bottom. It's CH with 2 CH3 branches. So let's have CH with 2 CH3 branches. So anyways, I'm going to do all the working here on top of this. I want to connect them together. So what would happen is, the OH and the H, they're going to be removed. The OH and H would be removed. The OH, they're going to link up. Okay, everywhere you're going to go, there's OH and H getting removed, and they would all link up. Okay, so these things are gone from the middle, and this carbon would then bond directly with the N on the other side. So that's an amide link. The same happens over here. The OH and H would be gone, and that's your amide link that's formed. Okay, they would be slightly closer together. So amide and okay, so you have all these amide links that are forming, And that's your polypeptide chain, and this is the sequence in which they're going to connect. So basically, you have these C double bond O and NH linkages. So these are your linkages, which are known as your peptide linkages or amide linkages. There's no difference between them. ठीक है? Ali, is this clear? Sara, is this clear?
Yes.
So that's so ठीक है, that's your primary structure. So the primary structure only tells you, and you'll be asked a question very similar to this, that you would be given a sequence of amino acids and you would have to draw a polypeptide chain. The next thing is that the next thing is known as your secondary structure. What's your secondary structure? The secondary structure is now like a strand, like a thread, like a piece of thread that's coming out of a spool. Uh, the the strand is not straight. It's not always straight. Uh, it gets entangled. So the secondary structure is that the primary structure folds and gets entangled with itself. And why does it fold? Due to hydrogen bonding. So due to hydrogen bonds forming between amide groups. So what's happening is that your chain had a lot of amide linkages. You can see above there were a lot of amide linkages. So what happens is that the two types of structures that are formed. The first one is known as a beta pleated sheet. What happens in a beta pleated sheet is that it's a flat sheet. The strand arranges in a flat manner. And why does it arrange? Because the chain regularly has these amide linkages. There's a serial bond O, and then there is an NH. So whatever the strand is, so these linkages are continuously appearing in the chain. And the same happens over here as well. So they're constantly appearing. And these NH bonds, they are highly polar. The H is strongly positive. And the oxygen over here has lone pairs. So if it has lone pairs, it's going to get attracted to the positive H, and there would be hydrogen bonds between these groups. H bonds are going to be formed, and they would be formed everywhere. So let me just probably Google a better version of beta pleated. So this is how it's going to look like. Probably this is a better way of looking at it. So the strand would be curling up, and there would be constant attraction between the strands because of the hydrogen bonding that's happening between the strands. Okay, is this clear? Saru, clear? Iba, is this clear?
Yes, sir.
Yes, sir.
अच्छा उसके बाद the other thing is there's another one which is alpha helix. Now in alpha helix, uh, the structure is not sheet like, it's actually helical. So it's a helical structure, and this one is known. पहले इसका नाम लिख लेते हैं कि this is your beta pleated sheet. And this over here is your alpha helix. The reason is the same. Uh, the reason is that there is serial bond O and NH as part of the chain. And there might be a serial bond O over here. and an NH over here, and the oxygen, which is partial negative, has lone pairs, is going to be attracted to the positive H, and there's going to be attraction. So you would have to just show that, draw that. So that's what an alpha helix looks like. So let's so this is one, probably this one. This one is a better. So the chain is actually curling up, but in a helical manner. And the top serial bond O and NH, the groups, they're all attracting each other. So that's what an alpha helix is. I said, what an actual protein looks like is that an actual protein contains both. It's not just beta pleated sheet or because the strand is very long. Okay? So if the strand is very long, it's going to curl up in many different ways. So an actual protein would look like this: that it would be forming beta pleated sheets, and then might start forming an alpha helix. Then might start forming these beta pleated sheets again, and then might start, then probably not do anything, then forms beta pleated sheets and forms alpha helix. So an actual protein is basically a combination of both things. So this is what actual protein, a protein chain would look like. Both alpha helix and beta pleated sheets would be present. Both of them are present. Now the other thing about this is that alpha helixes, they usually, so alpha helixes are usually more. I mean, the ones that look like springs, they're kind of more flexible, whereas the beta pleated sheets are kind of more rigid. So that would create two different properties for the protein. Uh, Alpha helix is more flexible, more spring-like, more stretchable, and beta pleated sheets are less flexible. So if you're talking about, let's say, spider silk is very stretchy. That probably indicates there's a lot of beta alpha helixes in the protein structure. Um, the human ear is not stretchable, so that means there's more beta pleated sheets in the structure. So that both of them, they kind of give the property that whether it's going to be the human skin is very uh, stretchable, so that means probably more alpha helixes. Uh, your nails, they're kind of very rigid, so that's more beta pleated sheets. Okay, clear hai, Ali? Is this clear? Abu,
Yes,
clear hai?
sir.
Yes.
And finally, the last one is that's your tertiary structure. And the tertiary structure is that even the secondary structure, the tertiary structure, there's going to be more folding, there's going to be more entanglement. So the tertiary structure is that your secondary structure folds or entangles further. So your secondary structure folds and entangles because of R groups, because of interaction. So you had all these R groups coming in, which we initially did not care about. Like you had a carboxylic acid, you had carbon chains, you had another carboxylic acid. So you have all sorts of things in the R groups. So if you have, let's say you have an alpha helix, Now, what's going to happen to the alpha helix is that the alpha helix would have lots and lots of groups that would be, so there would be lots of groups coming, and that would further fold the alpha helix. So it's not going to be just one straight spring-like structure. It's going to be further twisting around. Why would it happen that there might be an R group that would be coming out of the chain from one side, and another protein chain would have another R group, and they would start interacting with each other. Now, these R groups can have all sorts of bondings. I mean, every imaginable interaction is possible. There could be van der Waals forces between them because you had a variety of R groups. You could have permanent dipoles attracting each other. You can have hydrogen bonding between them. And you can also have ionic bonds and covalent bonds as well. So I'll just give you an example of ionic bonds. For example, there's an R group. Let's say there's an R group coming, and the R group is NH2. That R group is probably present in, I'll draw this over there. So that R group is, I mean, there was one R group which had NH2 in it, and that was lysine. So lysine had an R group which was CH2 4 times, and then there was NH2. So LYS. So the chain would have a side chain which would be your R group, and the other one can have you can have an aspartic acid CH2 COOH. So ASP, aspartic acid. One of them is a base, and the other one is an acid. So the base would gain an H plus 1 ion, so it would form NH3 plus 1. The acid would lose its H plus 1 ion, and it would become minus 1. So there's positive charge and there's negative charge, and there's going to be an ionic bond that would be formed between them. So they're going to have an ionic bond. You can also have covalent bonding, like cysteine has CH2SH, and there could be another cysteine residue in R group that could have CH2SH. The H between them goes away, dehydrogenation happens, and the sulfurs they end up bonding with each other, and they end up forming a covalent, and this is known as a disulfide bridge. So this is cysteine, and from the other side there's also cysteine. But if you so you can think of any attractive force between them. There are all sorts of R groups that are coming out of the chain. So when folding happens, the R groups they start interacting with each other, as you saw over here. In the original chain, the chain is going straight, and there's a lot of R groups that are coming out. So when the chain starts to fold and curl up and entangle, the R groups they would start bonding with each other, and all sorts of intermolecular forces or bonding can happen between them. And therefore, what happens is, if you let's focus on this structure. If you look at this structure, what basically happens is that the R groups they start interlinking. I mean, there's an R group over here, an R group over here. They would start interlinking with each other. So all sorts of interactions or bonding might be possible between these R groups. Okay, is that clear? Ali, is this clear? Sarah, clear? Aisha, is this clear?
Yes, sir.
Ripa?
Yes.
Yes, sir.
So you're done with primary structures, you're done with secondary structures, and you're done with tertiary structures. There are quaternary structures which are not in your course. The quaternary structure is simply that many different tertiary. I mean, a normal cell is a combination of many proteins. So you get these tertiary structures that one protein chain could be in this way. So you could have a combination of different tertiary structures. They could all link up to each other. And so you'll be asked about all of them. At the end of the day, a globular protein is usually formed. and it's going to have active sites, which are going to interact with other molecules and which are substrates. And you would have the lock and key model example of how a protein works. Okay, the lock and key model is this one. So it's going to be this thing. That at the end of the day, a lot of your protein structures, they're going to be globular protein, and they're going to be very shape specific. All proteins are very shape specific. They have, I mean, it depends on how the protein entangles and uh, uh, folds and forms primary, secondary, and tertiary structures. Uh, but at the end of the day, you're going to get different shapes, and there would be active sites. There would be sites on the protein, this complicated protein molecule, where different molecules called substrates can combine or react with. There might be R groups on this side that would be interacting with this molecule, and they would act on the substrate, and a reaction would take place. So this is your lock and key model. You must know how that's going to work, and that would make proteins very, very shape specific. Okay. So that's we're done with proteins, primary, secondary, and tertiary structures. Okay, we'll continue with the papers. We're almost done with organic now. Okay, there's only he's talking about zwitterine. We're going to do zwitterines in the next class then. Okay, so we'll continue with
Okay,
this.
sir.
कोई question नहीं है? Clear है सारों को? अरीबा clear है? Sarah, is this clear?
Yes, sir.
Yes, sir.
Yes, sir.
चलो ठीक है. Let's continue in the next class.
Okay, फिर अल्लाह हाफिज़.
Okay, take care. Allah Hafiz.